you hear me? Okay, perfect. So my talk will be in English, and I'm here today to talk about high content analysis and how we can use it to screen CRISPR libraries. But before I go a little bit into the technology, I want to start off with a little story from my PhD. So this image is actually from my PhD work, and it took me hours in front of the microscope to actually take the perfect image for the cover page. But it took me even more weeks to make the perfect sample before I started going to the microscope. So then my supervisor said, we want to have a good image to represent your work. But again, the whole work would have been nothing without actually the data and the results that come from those images. So what you have to keep in mind is that we can have all those nice images, but without the data that we can extract from the images, we cannot tell our story. We cannot convince someone that our research is really what we think it is and how we envision it. So that's why, in that time, if I would have known that there's a technology that can help me to extract those data from those images, I would have been lovely to take it. So that's why today I want to talk about the image cytometry. So what does it mean? Well, cytometry as such means we measure characteristics of a cell. And now in high content screening, and this is a sentence I would like to take home from this presentation, is we try to understand phenotypical changes of a cell, or characteristics of a cell, and not only a single cell, but a whole population of cells, by analyzing the images with certain algorithms. You could also say it's automated microscopy with the attached or subsequent analysis. Now, I use the terms high content analysis and high content screening interchangeably because for us, it's basically the same thing. And why do we think as Thermo Fisher Scientific, a commercial partner, helps you to do your research, that we have good expertise in this? Well, in 1989, we actually developed the first high content screening instrument on the market, where we said we want to have a microscope that not only acquires the images, but in real time actually analyzes them and gives you the graph afterwards. We have the most placed instruments, we have lots of publications, well over 1,000 publications, and we have a good product range for products which help you to get into high content screening, so they are very affordable, up to the really powerhouse that are mainly used in big pharma companies to screen for compounds in the drug discovery field. Now, at Thermo Fisher, we can also supply the fluorescent reagents that you would need to make something visible in the cell. And this brings me to my next point. What do you need to do high content analysis? Well, I say it starts with the reagents, because we need to see the certain parts in the cell that we want to analyze. So we can provide this by the molecular probes. And then comes the instrument, so actually the microscope that takes the images. But it doesn't end there. You have to keep in mind you need a really strong and powerful software that then can take those images, analyzes them and convert them into graphs and data so that you can understand what your population of cells is actually doing, right? And because we take not only a few images, we take thousands of images, we have to then also have all the IT and server capabilities afterwards to store those images and go back to track what cell was doing what. And I will come to that in the future. So in terms of the instruments, we think, why is high content and you just see a box sitting on a bench? What is it actually doing and combining? We say it combines three major technologies. One is microscopy, which provides us with the flexibility of having different samples, different magnifications. We visualize things in a cell, and we get spatial resolution of our sample, and we can localize certain things. But it also combines, for example, the screening capabilities and the automatization, the ease of use of a plate reader, because usually you work with plates in high content screening. And it combines also flow cytometry. Because in flow cytometry, you look at a single cell and what is it doing, how much fluorescence it has, but then you analyze again thousands or hundreds, thousands of cells, and you want to see what is the whole population doing, or how is the population different from one to the other. And that brings me to another point, to drive back my main message. What is high content screening? When I ask you, what's the difference between a zebra and an elephant? Well, one is a very large animal, right? It's gray, long nose. The other one is a little bit smaller with black and white stripes. So that is what high content screening is doing for you. It characterizes the cell based on certain characteristics, how they appear, and then analyzes it. Okay? So we can do certain multi-parametric assays, and they have been developed and increased over time really dramatically. So we have now, this is a little bit older graph from 2013. By now there are more than 1,000 publications that use that technology. And this is because we establish more and more applications where we can use it. For example, cell tracking, translocation, cell toxicity, proliferation, many different applications where you can use high content screening and use such a system to make the data. And because we are here today at a CRISPR conference, I want to talk to you that you can use those high content screening instruments or this technology also in your CRISPR workflow. Because CRISPR is changing the world, right? Everyone is talking about it, and it's a very powerful technique. But also, you have to somehow analyze your data or maybe your images. And with that, I want to go through a little bit of a screening workflow, how we envision it as Thermo Fisher, and where we can help you. So first, you select your editing technique. Why? Well, we are here with CRISPR. We can also use siRNA. And then we have a set functional test where you look either at proliferation, cell morphology, lantha screen, and cell sensor. I will come to that. And we have to optimize the delivery. So some of you have been at our booth, and we talked about how can I get my CRISPR construct into a cell. So there's electroporation, transfection, we can use viruses. And then after that, usually the part comes where I have a certain protocol that I need to validate. And I also want to automate the whole screening process. So I have a whole CRISPR library that I transfect, many different conditions. What's the fastest way or a good way to screen for hits? And this is where the high content analysis platforms come in. Okay, so now for example, where we use CRISPR and NF-κB screening, we have actually a cell line developed which is called cell sensor, and it has a beta-lactamase converts an enzyme or substrate in the cell which destroys the thread. So before the cells are green, or you can use green fluorescence to detect them, and when the NF-κB signaling pathway is activated, the cells become blue. So simply speaking, I mean, there's a lot more to this NF-κB signaling pathway, but under the stimulation with TNF-alpha, the cells should become green, right? Blue, sorry, and in the absence, they are green. So now, when we have our, let's say, CRISPR library, a lentivirus with CRISPR, and we transfect it into those cells, then we wait and add TNF-alpha, we can do a ratiometric assay and calculate how many cells are still green, a little bit more, blue, and become more and more blue, and then also have our controls. And the good thing is, not we have to do it by taking images at the microscope and counting ourselves by hand, which I have also done during my PhD. No, the instrument is actually doing everything for you. The plate goes in, and the data and the graph comes out, if you have set up the right parameters in the beginning. So there is another picture of what the system can do. It finds each and every single cell. It defines, for example, the nuclei, and then we can detect translocation of NF-κB, which is moving from the cytoplasm into the nucleus. And then the instrument can help you to track changes over time. And this is only one example. We can have many more. If we look at the cytoskeletal structure, which breaks down, how much is it breaking down, how far are cells migrating, how strong are they proliferating. All is possible with this technology. And this is why, this is my almost last slide, I want to emphasize, we have now an image which can give us a similar picture actually as a dot plot from flow cytometer. And the really cool part I love is that each and single dot is a single event in a cell from the image. So from those 1,000 images, I can click on one dot and it will show me exactly the nucleus from that cell. So it tracks back to the images that are stored somewhere on the server. And with that, I can of course create a lot more significant data and grasp high statistical significance. So in that sense, I usually say to customers, what can you do with it? Well, it's all about the data and your story. You want to tell a story and convince someone that your work is meaningful, and this you usually do by publications. And my doctor father says, well, publication is the currency of science. We need to write grants, but we also need those data, right? And that's why I want to end my talk with 5 main things I hope you get kind of and take with you. One is, what is high content screening? If I ask, it's looking at phenotypical changes of a population of cells based on image analysis. Okay? The other one is combines three technologies, basically microscopy, flow cytometry, and plate readers. And you need four essentials that we think before when you want to go into high content screening. So you need reagents, an instrument, a good software that detects everything, and the bioinformatics and IT support behind it. And then, well, I really love that the plate goes in, it acquires the images, and in real time, it's creating the data. So when the plate comes out, the graph is already done. So it's really, really powerful. And in CRISPR editing and screening, we also have lots of libraries developed on those platforms in-house where we do our own research at Thermo to develop those readout assays. And with that, I am happy to take questions or meet you at the booth to talk a little bit how we can maybe set up high content in your lab. Okay?
Thank you very much.